there I have the opportunity to develop a bioinformatics library using actually Faro. And so this talk is to present the work I did in that time. So as BioSmalltalk is the library for bioinformatics is part of the Open Bioinformatics Foundation, and of course it's a tool only to help. There are already hundreds of thousands of bioinformatic tools used every day by bioinformaticians, and so this is a kind of tool that I developed to kind of speed up the work I had to do using Faro. This is a bit of introduction because it's kind of, I know it's Friday, everyone is a bit tired, but we're going to talk about molecular biology. But no, I will try to make it easy. I'm not a molecular biologist or geneticist, but I will try to explain as I understood. So the area has a bit of context, which was the development of the Human Genome Project. And I want I really like this graphic because it shows what happened after the completion of the partial completion of the human genome, which I know that in the UI axis it shows the millions of dollars which cost at the point which was kindly a draft, and through the X we have the years that it takes. It actually started before, but at 2003 it was completed by 19%. But of course that people didn't know that it was partially completed. So and at some point in 2008 there was a drop, which was when the sequencing centers started to switch it to a new technology, which is called NGS, or for next generation sequencing. The previous one was called Sanger. And this development of a new technology really make the cost of sequencing really cheap. And the amount of data exploded, actually, because we passed from analyzing just one or a group of genes in an organism to analyze thousands of them. And this enabled to new areas like functional genomics, just to the functions of genes in a more contextual way with more data, like a kind of development. So, and at some point, this will impact in what's called personalized medicine, which is kind of when the technology became accessible to all of us at a really cheap cost, and this will be like another huge development of the technology. But this is like one of the driven factors through the years. So, but from by the way, the whole cost of the project just the Human Genome Project costed $10 billion. So that's the funding we were talking about. So, but to go to the other side, what's I really like this one because it summarizes well enough without too much complexity what's happening inside of us. So each cell in our organism contains a nucleus, and the nucleus has its own copy of the genome. And basically, the genome is all of our organism's DNA, and we could see that it's organized in chromosomes, of course. And DNA is a chain of small molecules, which are called nucleotides. And nucleotides coming, of course, that we represent with the 4 letters, which are A, C, G, T, and they came in pairs. in 2 strands. And if you, they came like always like A is paired with G, and C is paired, cytosine is paired with G. And if you ever need to remember this, you could use Alan Turing and Carl Gauss, because like it's easier to memorize. It's like a mnemotechnic rule, yes. And also, this is basically the structure and the kind of objects that we manage is at the very basic level is managing this kind of molecules. And for the more functional part is how does it really oversimplification, of course. How does it work? So we have 2 strands of DNA, and we have a process which is called transcription. And transcription is like essential to life. It's the first step of gene expression, and it consists of translating, copying the DNA into RNA, which is another kind of molecule. And this is like the beginning of the gene expression, and when the cell needs to do something, like it locates the right position at the DNA, and it starts to transcribe. And then there is another process which is called translation, and the translation is takes the RNA by 3, and this is like it's called a codon, and this translates into amino acids, and which together they build a protein. So of course that there is other type of RNA, like non-coding RNA, which has other type of functions like regulatory functions, and there are hundreds of, for example, non-coding RNA, but this is like a kind of basic overview. So we have in the library we could like represent these operations. We could use we start from a sample sequence, and we just convert it in an object, and the object has already, of course, it shows the sequence length and the DNA alphabets, and DNA alphabets as it has its own hierarchy of properties, because every kind of alphabet, like DNA or RNA, has different codes, and from there we could, like, from the sequence we could transcribe, and it has another kind of alphabet, which is RNA alphabet, and we could back transcribe, which is this is actually called complementary DNA, but for our case, the thing is that we have like a mismatch between what happens in vivo, in vitro, and in silico. So not all the operations which happens in real life in biology has like a sense to represent in the computer. So we are like selecting which operations that we perform. But of course, biology is way more complicated. Of course, we have the complement, the Alan Turing and Carl Gauss thing, and this like lets you represent the 2 strands to the complementary sequence. And we have, of course, the reverse complement, because the DNA is read in one strand, is read in one direction, and from the other strand is read in another direction. And finally, we can translate to the protein sequence. So as it's read by 3, you see that the protein, the amino acids are only 3 for that sequence. So we have also sequence utilities. We could calculate these are some sequence statistics, like the GC content, and the GC is a value that researchers look because DNA is the nucleotides, the 4 nucleotides is without selective pressures or any kind of mutational event. they are equally distributed in the genome. So when you have an overrepresentation of GC, of guanine cytosine content, you could have like a contamination of samples, and like that you could check if your sample procedure was correct. This is a measure that should be like really low standard, and we could of course plot with the help of Rosal. And we have another, because in the end this is all thermodynamics, and we could like get the molecular weight. There are algorithms for that, and the local complexity is another measure which lets to determine the variant nucleotide composition, and of course the occurrence of letters also to determine if the sequence quality. And we have also that because sequences nucleotides could be represented by there could be like many positions, because sometimes there are the sequencing machines, they give errors. You could have, for a position, you could have different possibilities. Of course, we have some algorithm for pattern matching, which is counting some subsequences, and the longer and some checksum measures, which are pretty standard in the bioinformatics world. And of course, that we could plot the distribution of the FASTA. FASTA is the basic format in bioinformatics. It's a plain text, and it was developed in the 70s, but it's still alive today. The thing is that, of course, we have a sequence alignment, but the thing that I want to tell you that you could see like FASTA format is one format if you ever like want to experiment this, but there are multiple formats in bioinformatics. It's not like an area which is like satellites or the super collider where you have just one type of data. Here you have millions of organisms, and you have also the instrumentation methods for each of the organisms, and each species has its particularities. And if you combine everything together, and you have different areas of study like transcriptomics, which studies the RNA, and you have genomics, which is focused in the DNA, and proteomics and metabolomics, and you mix all of that, and you have a very heterogeneous field with multiple databases. So it's not like you have a single format. You have a multitude of formats, and there are some kind of lack of coordination efforts to join these together. So it's very common. It's very complex because biological organisms are really complex. So this is one of comment I wanted to make. So sequence alignment is a big field in bioinformatics, and we have seen if you have seen the Rosal presentation, we have seen, and these are more for the biological explanation. These are kind of matrices to represent the history of the. Actually, they represent an hypothesis of the evolution of multiple organisms. On each line, it represents an individual, and each column is called a site, and it represents what happened in any specific position of a chromosome. And you could see that there are some gaps, and there are some regions that are more like similar, and those represent like conserved regions where the function of a gene was like positively selected by the evolution of a species. And there are some gaps where you have a very variant like mutation, and you could study why there is some mutations there, what happened in the evolutionary history. Of course, that you could build manually an alignment, But the thing is, like, mostly they are like, you could have a sequence of study, and you could make the query remotely, and like compare with different, like from nucleotides to protein, or nucleotides against nucleotides. You have different kinds of queries, and you will do that like this, local search, and you could use other software for doing alignment. This is the sample input for aligning the FASTA sequence of the COVID genome. This is all implemented with wrappers, because everything that is already researched. And we have also a local aligner implemented, which implements the Needleman, which is a technique of dynamic programming, which implements a variant of alignment, which is global alignment, which aligns end-to-end a sequence. There are other technique which used by BLAST, which is the local aligner, which only looks, given a wider sequence, only looks the specific part that you are interested. So we have an implementation of these algorithms, basically a matrix scoring. It uses a matrix scoring and traceback to optimize the problem of sequence matching, and we could like implement very more complex pipelines. For example, for retrieving all the sequences for the COVID, for example, we could just with the identifiers. By now there are 8 million of identifiers of sequences of the COVID genome, and we could align them and parse them just in a few lines of code. And of course, that we could have other we have other functionality, like getting consensus sequences, which is the most represented nucleotide at a position. We have repeats, like analyzing most of the DNA is repeated, and we don't know yet the function of this. It was called chunk DNA, but through the years we are realizing that it's not actually chunk, it's that we don't know what it's doing. And but yes, some type of it is called also fossil DNA, because it showed that it has some function in the past of our history, but now we don't know. And codon tables, which are useful for translating the protein, and this is all implemented in the following the nomenclature of UPAC, which is an institute of applied chemistry. And of course, I've shown the alphabet features and records are like more like high-level representations of the information of sequences. And we have also just one-liners to download genomes of the species that you want. The genomes are also versioned. Wrappers, we have wrappers for population genetics. Links and Catadapt is a tool for working with the next generation sequences, which is a lot way more data that represents the whole genome of an organism. And other BLAST, of course, and Cluster for multiple sequence alignment, and Structure for population stratification, like determining the structure of a population without previous knowledge, and have shape it for genomic imputation. And HH suite is for hidden Markov model profiling, like you could search with the algorithm, you could search the homology. The thing that with bioinformatics is that you use this transfer of knowledge from other species to know what your species, what your sequence is doing, like which function does it have. So you try to use to transfer the knowledge from other species to the one that you're studying. So there are many ways to do that, and one is sequence similarity, and the other is using hidden Markov models, which try to look for patterns in in regions of a sequence, and we have other aligners. And we have formatters to format all the multiple kind of data we could have, and FASTA format and GenBank and other formats for population genetics. And we have parser for all these, for XML and given by the GenBank and the NCBA and TRES. This is actually like a kind of point of entry to the library if you want to experiment once. Because the interest library, basically the whole thing is organized in primary and secondary databases. And the primary databases is where the place where researchers upload the sequences, like a kind of archive. And they perform some kind of basic annotations, but they are not really well created. It's somewhat created. And this is organized globally in 3 data centers. We will say that one is from the GenBank, which is located in the United States, and the EPA, which is here in Europe and United Kingdom. The DCA is from Japan. So these are synchronized every day, and they contain millions of trillions of nucleotides. Of course, that you have a multiple repository, and everything is accessible from the library, with just a kind BioInterest client. Like you could have access to all of these database. Like some of them are very specific, like cancer chromosomes or cancer bioinformatics or I don't know, for proteomics or taxonomies. So this is like a huge area. So this is kind of the high overview, level overview. And we have some applications that through the years, and in this case, for example, one application, the real application that we have used the library is to determine, this is complex to explain, so give me a second. So the idea is that you could have some breeds that are composed. Actually, this happens in animal genetics, and you kind of compose a breed to have a positive selection of some features. For example, resistance to heat in tropical areas. And so we wanted to know here the goal of this research was the particular region of the chromosome 23, which is called sorry, I forgot the name. The major histocompatibility index, which encodes for genes which they are suspected to have a response for adaptive immune response, and this is linked to resistance to tropical environments. Also, we determined the goal was to determine which was the ancestry which has most impact in the development of this Brangus cattle, which is a combination of Brahman and Angus. And so we came to the conclusion that Brahman was the founder that has most impact for this particular characteristic. And this was like a bit more fun, which there was a call from a prosecutor, and he has an hypothesis that the suspected was arrested from crimes. The victim has a dog, and the shoes of the suspected has some kind of fecal material, and the prosecutor hypothesis was to see if they match. So we kind of did a phylogeographic analysis with using these databases to help in the forensic case, in this particular forensic case. So we developed a kind of workflow for this kind of thing, which is now kind of in the public eye. It was in the public eye because this was like original to check, because from fecal material the DNA is very degraded and it's difficult to see to extract some information from there. And we have runs of homozygosity, which is kind of measuring the inbreeding levels, and this gives a measure of health risks in population. So in this analysis, it's a bit more complicated, but the analysis was that using this metric, which is called runs of homozygosity, which are inherited alleles that from one generation to another, that give some kind of indication of the possibility of having a population which is has more health risk because of inbreeding, which is mating of closely related individuals. So this, of course, there is the paper of the library BioSmalltalk itself, and we publish it, and we have some benchmarks to measure against other libraries, and we outperform for example, in the parsing of big XML files. We outperform other libraries. Recently, I migrated everything because this was done with Petit Parser 1, and now I'm using Petit Parser 2, which is pretty fast actually. Okay, so everything is available here in the repository, and thank you. Now, if you have some questions, it will be.
If I understand correctly, when you have those triplets of nucleotides encoding amino acids, the number of possible amino acids is less than all the possible combinations. So that means that if you have a sequence, you should probably be able to determine if you have it in the correct order, or you need to reverse it with high degree of confidence. Is it correct?
You are asking about if the translation from RNA to protein.
No, you have a sequence of DNA which encodes amino acids.
Yes.
And you have only 12 amino acids, which means that some of them do not make sense. So if you have a sequence and then you see that there is more combinations that make sense than those that do not make sense, and if you reverse it, it's in opposite, then you may be quite sure that you have the sequence in the right order. Is it correct?
Actually, with the thing with the comparative genomics, like comparing sequences, which is kind of the part of the alignment of sequences, is to determine what's called homology, and when like you see what's for each position is like it's not that it doesn't make sense, but you need to reaccommodate the ordering, so to see, because of the sampling method and the procedure of getting from the material the DNA material to the machines is like very prone error, and could have like multiple errors of lectures of reading. And from this, it's not that doesn't make sense, but the most part that you need to like realign this, like to see which site has more homology, like it's like common ancestry information with one which another. So so it's kind of that you need to start with a hypothesis of what you are looking for. So this is why you align specific regions against more complete annotated or reference genomes. This is the point where most people start. So this is very, for humans, it's really well annotated. So if you are going to study some human disease or everything, it has each position is really well annotated, and there are lots of data supporting, especially where where there is positions where there are mutations. For animals, it's more complicated because there are not so much funding for them. There are actually a lot, but not so much as human. So, and especially when the mutations related with cancer, for the cancer bioinformatics is they're getting the most funding actually. But yes, it's kind of, that is kind of the point of start. Okay.
In bioinformatics, there is a lot of uses of computational notebooks.
Yeah.
At least with Python, for example,
Yeah.
Python and stuff.
Yeah, I know.
I don't know if you have experience with teaching or creating, let's say, educational material, and how do you see the use of computational notebooks in this case, for example, for BioSmalltalk, or what's like kind of the picture of using computational notebooks in bioinformatics using Smalltalk?
Yeah, the area that you're asking, I think that you're asking is about exploratory bioinformatics, which is kind of different of confirmatory. But when you're doing exploratory, what I've seen of using notebooks like Jupyter and everything is that people usually start like managing, but when the workflow diverge, like you need to really need to do multiple parallel experiments, but because you are like exploring and you need to have a different workflow path. It doesn't scale well for what I've seen. So this is why I really like it to use Faro, because you could have multiple inspectors and you could see at the same time what's happening. And this is with the sequentiality of notebooks. You like end up scrolling because this workflow is really long, like really long, and you can finish in 1 week. Like it could take a month. Actually, and it was a problem to share this notebook, because for one person it was okay, but when they wanted to share to other, they like, no, you need to execute this part and then this part and this part. So it was like the workflow was, the ordering thing was like a need for communication, verbal communication, and it didn't scale for what I did. So the idea of the library, it was like kind of provide a different way to develop this kind of things. Okay. Thank you.